You know that feeling when you're trying to carry like all the groceries inside in just a single trip?
Oh, yeah, the classic one-trip challenge.
Right. You've got bags hooked on literally every finger. The plastic is just, cutting into your skin. You are almost at the front door, and suddenly your hands just start opening.
Oh, you'll just give up.
Exactly. It's not a conscious decision to let go, right? Your brain is screaming at your hands to hold on, but your muscles are just completely ignoring the command. It feels like this biological betrayal, like
Sure.
mechanical failure happening out on the very edge of your nervous system.
It really is a profound disconnect. I mean, you are generating the conscious willpower, right? You're sending the neural drive down your spinal cord, and the tissue at the end of the line simply refuses to execute the order.
And for decades, if you asked like a gym teacher or a fitness trainer why that happens, you'd get this deeply confident, incredibly simple answer.
Oh, absolutely. The classic lactic acid explanation.
Yeah. They would tell you that your muscles just ran out of oxygen or that lactic acid was building up and, burning the muscle out. But today, we are looking at a fascinating stack of anatomical and physiological research on muscle fatigue and oxygen debt to completely dismantle those assumptions.
Because the reality is so much more complex.
So much more. The goal of this deep dive is to really unpack the actual biochemical reality of what happens when human tissue is pushed to its absolute limit for you, the listener. Because as it turned out, a lot of what we consider just basic common knowledge traces back to a severely misinterpreted Nobel Prize from 1922.
Which is wild to think about. But to really understand why a piece of machinery stops, we have to start by looking at what makes it go in the first place, right? The fuel source.
Okay, let's unpack this. Where do we start?
Well, from first principles, every single muscle contraction, I mean, whether it is blinking your eye or deadlifting 400 pounds, it all runs on one specific molecule, ATP or adenosine triphosphate.
Ah, right. The classic biology class powerhouse.
Exactly. But the actual mechanics of how it provides energy are fascinating. You have this adenosine molecule with a tail of 3 phosphate groups, right? And the bonds holding those 3 phosphates together are highly unstable. They hold immense potential energy.
Okay.
So when the muscle needs to contract, an enzyme just cleaves off that 3rd phosphate. That chemical bond snapping is what actually releases the energy to power the muscle. And that leaves you with ADP adenosine diphosphate and a loose inorganic phosphate molecule floating around.
Right, so it basically breaks off a piece of itself to create an explosion of energy.
Yeah.
But there's a catch with this, right?
A huge catch. It's the storage capacity. A working muscle fiber only stores enough readily available ATP for roughly, 1 to 3 seconds of maximal effort.
Wait, just 1 to 3 seconds?
Yeah, that's it. If you relied solely on the ATP currently sitting in your forearm muscles, your grocery bag trip would end before you even got the trunk of your car closed.
That is wow. So everything that happens after those first 3 seconds is just a frantic cellular race to regenerate ATP just as fast as you're burning it.
Precisely. It's like having a tiny amount of cash in your pocket, but you're spending it constantly, so you need a massive, immediate income stream. And the body tackles that crisis using 3 distinct energy systems, and each one has a very specific failure point.
Okay, so what is system 1?
System 1 is the phosphocreatine system. So muscle cells keep a reserve of a molecule called phosphocreatine. When you burn that initial ATP and you're left with ADP, phosphocreatine basically steps in and saves the day.
How does it do that?
It immediately donates its own phosphate group to the ADP, converting it right back into ATP.
Oh, so it just patches it right back up.
Exactly. And this happens instantaneously, and crucially, it happens completely without oxygen.
Wow. Okay, so that is incredibly fast. But the source notes that the reservoir for this is tiny. This phosphocreatine buffer only buys you about 8 to 10 seconds of all-out effort, right?
Right. And the perfect biological example of this, the absolute limit, is Usain Bolt's 100-meter dash.
Which he ran in 9.58 seconds.
Exactly. His world record is mathematically and biochemically the absolute outer limit of this first system. He ran that race almost entirely on phosphocreatine turnover.
So it's basically the ultimate biological sprint. But what happens at 10 seconds when that cash runs out?
Well, the cell faces a severe energy deficit. It has to pivot immediately to system 2, which is anaerobic glycolysis. Your cells begin breaking down glucose, so taking sugar from the blood or glycogen stored in the muscle and metabolizing it without oxygen.
And this churns out ATP really quickly to meet the demand, right?
It does, but it is incredibly inefficient. And it generates a massive amount of metabolic byproducts, specifically pyruvate, which quickly converts into lactate.
Right, okay. And this anaerobic system covers efforts lasting from that 10-second mark up to about 2 minutes, right? Because reading the physiological data, it highlights that athletes competing in the 400-meter or 800-meter track events report the most excruciating, agonizing fatigue of any human sport.
Oh, they absolutely do, because they are trapped in this brutal middle ground. The event is too long to run on the instant phosphocreatine buffer, but it's over too quickly for the body's third energy system to fully engage.
So they are forcing the muscle to operate almost entirely in that highly inefficient byproduct-heavy state. That sounds awful.
It is punishing on a cellular level. But then you have system 3, right? Oxidative phosphorylation. This is the end game. This is where the mitochondria take over for anything lasting beyond a few minutes.
So this is where oxygen finally enters the chat.
Exactly. The mitochondria start oxidizing glucose and fatty acids. This requires a steady supply of oxygen, so it produces ATP much more slowly than the first two systems, but it can run almost indefinitely.
Right, so the ceiling here is entirely based on fuel availability.
Yeah.
And your VO2 max, like the maximum volume of oxygen your heart, lungs, and vascular system can physically transport to the tissues.
Spot on.
But wait, synthesizing those three systems reveals something that completely changes how we talk about being tired. Because fatigue isn't just one thing, is it? Like, the fatigue a sprinter experiences is chemically a completely different reality than the fatigue of a marathon runner.
It really is. A marathon runner operates in System 3. When they hit the infamous wall at, say, mile 20, their mitochondria haven't failed. They have simply run out of stored glycogen.
So they just literally ran out of physical fuel.
Exactly. But a sprinter's fatigue is driven by emptying that tiny phosphocreatine reserve. We use one word fatigue to describe wildly different states of cellular crisis.
That is wild, because in those endurance events, it's a fuel depletion problem. But in those shorter, high-intensity events, the muscle still has plenty of glucose available, but it physically stops working anyway.
Right, the fuel is there, but the machinery jams up.
Which brings us to the actual fibers. If a marathoner just runs out of gas, what is physically jamming the gears of the muscle in those excruciating 2-minute anaerobic events?
This is where we dive into the cellular level. The research on peripheral fatigue, specifically drawing from landmark reviews by Robert Fitz in 1994, and Allen and colleagues in 2008, defines this failure not as a sensation, but as a measurable drop in the muscle's mechanical ability to generate force.
The physical machinery is compromised.
Yes, and to understand that, we have to look at the cross-bridge cycling.
Right, the mechanical failure. So, just for everyone listening, muscle contraction happens when thick protein filaments called myosin reach out, grab onto thin protein filaments called actin, and pull.
Exactly. But the myosin can't grab the actin unless calcium is released into the cell to expose the binding sites on the actin.
And that calcium release is basically the trigger for every movement you make.
But remember how we discussed ATP breakdown? Snapping off that third phosphate leaves behind loose inorganic phosphate molecules. During intense exercise, the cell rapidly fills up with this inorganic phosphate.
Okay, and reading through the cellular mechanics and the sources, the best way to visualize this, for you listening, is like a game of musical chairs. The binding sites on the actin filaments are the empty chairs, right?
It's a great way to think about it.
And the calcium needs to sit in those chairs to allow the proteins to grab on and contract the muscle. But the inorganic phosphate molecules flood the room and sit in the chairs first. They physically occupy the binding sites, blocking the calcium from doing its job, which literally prevents the proteins from grabbing each other.
Yes. That structural interference is one of the primary drivers of force loss. The muscle is essentially choking on its own exhaust. And on top of that physical blockage, you have a severe electrical problem involving potassium.
Because neurons and muscle cells rely on a voltage gradient to send signals, right? The cell pumps sodium out and potassium in to create an electrical charge.
Right. And when the brain sends a signal to contract, the gates open, and potassium rushes out of the muscle fiber to generate the action potential. Under normal conditions, a sodium-potassium pump pushes the potassium back inside to reset the voltage.
But during those rapid, repeated contractions, the pump gets overwhelmed.
Exactly. Potassium begins to pool in the extracellular space just outside the muscle fiber. And this disrupts the voltage gradient, drastically reducing the excitability of the muscle membrane.
So the nervous system is basically screaming at the muscle, firing the signal at full volume, but the electrical impulse gets muffled before it can even trigger the contraction.
Yes. And the cascade continues into the calcium storage tanks themselves. The sarcoplasmic reticulum, which houses all the calcium, starts malfunctioning. It releases less calcium with every muffled electrical impulse, and it vacuums the calcium back up much more slowly.
Okay, so let me summarize this for the listener, because it's not just a fuel problem. It's an electrical problem with the potassium, it's a signaling problem with the calcium, and it's a mechanical jamming problem with the phosphate.
That is the perfect triad of peripheral fatigue, yeah.
But wait, the glaring omission here is the burn.
Ah, yes, the burn.
Because I was always told, and I think everyone listening was always told, that the burn was hydrogen ions dropping the pH of the muscle from 7.0 down to around 6.4, and that acid is what stops the muscle from working.
Right. Well, acidosis absolutely occurs, but decades of exercise physiology placed far, far too much blame on it. Modern isolated fiber studies have demonstrated that at normal human body temperatures, this pH shift actually has a surprisingly small direct effect on the mechanical failure of the muscle.
Really? So the acid isn't actually stopping the muscle?
Not directly, no. The acid is acting more like an alarm system than a physical brake. It stimulates the pain-sensing nerve fibers, creating that agonizing burning sensation to warn the brain that the muscle is in a state of high metabolic stress. But it isn't the primary compound jamming the mechanical gears.
Wow. And that realization requires us to tackle the most deeply entrenched myth in the history of human fitness, right?
Yeah.
The relationship between lactic acid and the oxygen debt.
Yes, because the origin of this myth goes all the way back to 1922.
Right, A.V. Hill.
Exactly. A British physiologist named A.V. Hill, working with Hartley Lupton at Manchester University. They were strapping these primitive, massive gas collection bags to runners. They were measuring respiration and noticed that long after a runner finished a grueling sprint, their oxygen intake stayed significantly elevated.
So they were panting heavily for minutes after the work was done.
Right. And Hill theorized that during the intense anaerobic portion of the sprint, the runners had borrowed against their bodily oxygen supply. He called this the oxygen debt. He proposed that the heavy breathing afterward was the body consuming excess oxygen to repay that debt, specifically by clearing out the toxic lactic acid that had built up in the blood.
And concurrently, a German biochemist named Otto Meyerhoff was mapping the exact cellular chemistry of how glycogen breaks down into lactic acid without oxygen. And the scientific community basically merged Hill's oxygen debt measurements with Meyerhoff's chemical pathways, right?
They did, and it won them the 1922 Nobel Prize in Physiology or Medicine.
That is just wild. Think about that. A casual sentence in a high school textbook about lactic acid and oxygen debt was actually cutting-edge, Nobel-winning science from the dawn of exercise physiology.
Yeah.
But it was wrong.
It was. Well, science is rarely a straight line, you know? It took until the late 1970s and 1980s for a physiologist named George Brooks at UC Berkeley to deploy precise isotope tracing technology tools that A.V. Hill could only dream of to track exactly where that lactate was going inside a living body.
And I genuinely struggled with this part of the research when we were preparing because the paradigm shift is just so massive. Brooks proved that lactate is not a toxic, dead-end waste product. It is a highly valuable, high-octane fuel.
It is. He formalized the lactate shuttle hypothesis, showing that the body actively transports lactate out of the working muscle and sends it through the blood to the heart, the brain, and the liver.
Wait, so it's feeding the vital organs?
Yes. Think about the biological logic. The working bicep or quadricep is operating anaerobically because it cannot get enough oxygen. It produces lactate. But the heart and the brain are highly vascularized organs with massive concentrations of mitochondria and abundant oxygen. They can take that circulating lactate, oxidize it immediately, and yield massive amounts of ATP.
That is unbelievable. So the working muscle is essentially drowning, but it manages to manufacture a premium fuel and throw it as a lifeline to the vital organs to keep the organism alive during a crisis.
Exactly. And the liver even takes a portion of that lactate and converts it right back into fresh glucose to send back to the working muscle.
So Brooks completely demolished the foundation of the oxygen debt theory. Because if lactate is a premium fuel that is actively consumed by the heart and brain, the body does not need to consume a massive surplus of oxygen simply to clean it up like toxic waste.
Precisely. Which is why exercise physiologists abandoned the term oxygen debt entirely. They replaced it with EPOC, excess post-exercise oxygen consumption.
EPEF. Okay, so that explains what all that post-workout panting is actually accomplishing. The extra oxygen isn't clearing acid. It's driving a series of critical recovery processes. It's rapidly regenerating the phosphocreatine stores in the muscle, offsetting elevated core body temperatures, reoxygenating the blood, and balancing hormones like adrenaline.
Right, and the lactate itself. It returns to baseline resting levels in the blood within 30 to 60 minutes after you stop exercising. It clears incredibly fast.
Okay, I have to stop you there and play the role of the skeptical gym goer. Because what about next day soreness? Literally everyone calls that lactic acid buildup.
I know, everyone does, but the timeline makes that biochemically impossible. Delayed onset muscle soreness, or DOMS, peaks 24 to 72 hours after the workout. The lactate has been gone for a day and a half.
Wow.
That extreme soreness is actually caused by microtrauma?
Oh, right. So specifically, eccentric movements, like the lowering phase of a heavy squat, cause microscopic tears in the muscle fibers and the connective tissue.
Hmm.
And the body responds to that structural damage by triggering a localized inflammatory response.
Exactly. Immune cells rush to the area, swelling occurs, and the tissue begins remodeling itself to handle the load better next time. The pain is the sensation of inflammation and repair. It is completely disconnected from metabolic acid.
Okay, so if the muscle isn't poisoned by acid, and we know exactly how the local biochemistry fails with the phosphate and potassium, is that the whole story? Or is something else pulling the strings?
This is where it gets really interesting. Because treating the muscle as an isolated machine ignores the most complex variable in human physiology, the central nervous system.
Right, the brain. So the debate shifts from peripheral fatigue happening out in the limb to central fatigue happening in the brain and spinal cord.
Yes. In the late 1990s and early 2000s, a South African physiologist named Tim Noakes proposed a highly controversial model called the central governor theory. Historically, fatigue was viewed as a bottom-up process. The muscle runs out of ATP, the pH drops, and the tissue fails catastrophically.
But Noakes argued that human physiology is too elegant to allow for catastrophic tissue failure. He proposed that fatigue is a top-down anticipatory regulation.
So you're saying our brains hit the brakes before our muscles actually run out of gas?
Exactly. The brain is constantly monitoring the feedback loops. It senses the dropping oxygen levels, the rising core temperature, the accumulation of metabolic byproducts, and long before the heart muscle suffers ischemic damage from lack of oxygen and water, the bicep literally tears itself off the bone. The brain deliberately reduces the neural drive to the working muscles.
Wow. It forces you to feel exhausted and slow down to protect you. Yes. And the empirical evidence for this subconscious regulation isn't just theory. It's measurable via an experimental technique called twitch interpolation. It reveals the hidden reserve capacity the brain refuses to let us access voluntarily.
I love this experiment. The setup is just fascinating. So for the listener, you place a subject in a lab and have them squeeze a grip strength dynamometer. You instruct them to pull with absolute 100% maximum voluntary effort. Like they are consciously trying to recruit every single motor unit in their forearm, and they physically cannot generate another ounce of force on their own.
Right, they are totally maxed out mentally.
But while they are at their maximum voluntary limit, the researcher applies an electrical shock directly to the motor nerve of the arm. And what happens?
When that electrical stimulus hits the nerve, the muscle twitches and produces a measurable spike of additional force.
It produces more force, meaning it proves that the muscle tissue itself was not completely tapped out. The biochemical capacity to contract harder was still sitting right there in the fibers, but the central nervous system simply refused to fire the highest threshold motor units.
It kept a safety reserve. Now, Noakes' central governor model sparked fierce debate because it challenged the pure cardiovascular and peripheral limits established by A.V. Hill decades earlier. But today, the field of exercise physiology basically operates on a synthesized model.
Right, fatigue is multifactorial.
Exactly. It's a mix, an incredibly complex interplay between the peripheral biochemical limits of the tissue and the active anticipatory regulation of the central nervous system.
Grounding all of this cellular chemistry back in reality completely changes how we approach physical performance. This isn't just trivia, like the endurance world is built around managing System 3, right? Marathon runners carbo load with pasta the night before a race and consume concentrated glucose gels every 45 minutes, specifically to delay glycogen depletion.
Right. They are trying to push back the moment the mitochondria literally run out of substrate.
And then on the other end, high-intensity interval training, or HIIT, relies entirely on manipulating System 1 and EPC.
Yes. A typical protocol involves a 10-second all-out sprint followed by a 2 to 3 minute rest period. That specific recovery window is dictated entirely by the biochemistry of phosphocreatine.
Oh, because it takes the body a few minutes using the excess oxygen of EPOC to fully regenerate the phosphocreatine pool so you can achieve peak power again.
Exactly. And, understanding EPOC also deflates a lot of fitness marketing. The industry heavily promotes the afterburn effect, promising that an intense 20-minute workout will keep your metabolism elevated for days as you burn off massive amounts of calories.
Right, the magical afterburn.
Yeah.
But the physiology shows that while the body does consume extra oxygen to restore homeostasis, the actual caloric expenditure of EPOC is far, far more modest than the advertisements suggest.
Unfortunately, yes. But expanding beyond athletics, this biochemistry is fundamental to medicine. Conditions like chronic fatigue syndrome and various mitochondrial myopathies involve pathological dysfunctions in these exact same energy pathways.
Right, so by meticulously mapping how healthy muscle tissue fails under extreme stress, researchers gain the tools to understand and treat metabolic disorders where the system fails prematurely during just normal daily tasks. The biochemistry of an Olympic sprinter pushing through the final meters of a race and the daily physiological reality of someone managing a metabolic disorder trace back to the exact same mechanisms of calcium, potassium, and ATP.
It's all the same fundamental biology.
Which brings us right back to the front door with those heavy grocery bags. You, the listener, the next time your fingers start uncurling against your will, you aren't just experiencing a random failure. You are witnessing a beautifully complex biological cascade. The inorganic phosphate is flooding the musical chairs to block the cross bridges. The potassium is muffling the electrical signals at the membrane, and your central nervous system is stepping in as the ultimate governor, gently but firmly prying your fingers open to protect you from damaging your own tissue.
It's a perfect defense mechanism.
It really is. And consider the implications of the twitch interpolation experiment we just discussed. If a simple, localized electrical shock in a lab can bypass your absolute maximum voluntary effort, forcing your muscle to contract harder than you can consciously make it, who is actually in control of your physical limits? Is it your conscious willpower and determination? Or are you operating within hidden neural safeguards, safely locked inside a cage of your own protection? Thank you for exploring the physiological reality of fatigue with us. We will see you on the next deep dive.